I am Matt Pillar, host of the Business of Biotech podcast, chief editor at Bioprocess Online, and your host for this Bioprocess Online Live. I, too, would like to thank Cytiva for its support of today's program. And it's an important one given the disruptive advance of, or resurgence, if you will, of antibody-drug conjugates in the biotech landscape. Conceptually, ADCs have been in development for 100 years, but there are many reasons that the concept has largely faltered, and most of those reasons can be traced back to monumental manufacturing challenges. Throughout today's discussion, we're going to dial in on some of those challenges and how our guests in a growing industry of ADC developers are addressing them. Joining me for today's talk are two women on the front line of the ADC development effort, and one man who's got a unique bird's eye view of it all. Dr. Amy Johnson is VP of Medical Affairs at Vincerx Pharma, the ADC Upstart, founded by Drs. Ahmed Hamdi and Raquel Izumi, that's got a phase 1 ADC being tested in leukemias and MDS, a preclinical ADC in B-cell malignancies, and a handful more in discovery for solid tumors. Dr. Johnson was previously Senior Director of Translational Research at Janssen Oncology and Associate Professor of Medicine at Ohio State University, where she led an experimental therapeutics research lab, the university's leukemia research program, and served as co-principal investigator on multiple National Cancer Institute grants. Sandra Poole is Chief Operating Officer at the Dr. Brian Fisk-founded Mythic Therapeutics, whose development platform has produced an ADC candidate currently enrolling for a trial in non-small cell lung cancer. Sandra was previously COO at Logic Biotherapeutics and EVP of Tech Ops at commercial development at Immunogen. And before jumping into biotech, she earned her stripes in commercial manufacturing management at Genzyme. She's on the board at Travere Therapeutics and was formerly on the boards of ViaCyte until its acquisition by Vertex and Valneva. I mentioned the founders of those two companies, by the way, because Drs. Hamdi and Fisk have both been guests on my Business of Biotech podcast, episodes 156 and 203. Shameless plug. If you'd like to learn more about Vincerx and Mythic, I encourage you to give them a listen at bioprocessonline.com or anywhere else you listen to and subscribe to podcasts. Finally, Dr. Joe DeKosh is the best color man in the ADC game because he's the best student of the ADC game. Joe is a therapeutic practice lead at the market intelligence agency DeciBio, where he is the unparalleled expert on the ADC market. I'm really building you up here, Joe. He studies who's doing what, when, and where. He charts it, he monitors it, he graphs it, and he reports on it every day of his life. Joe, who holds a Harvard Ph.D. in immunology, has been an invaluable resource to Bioprocess Online's coverage of the ADC space, and he's doubling down on that commitment today as he takes a break from a vacation he's on to join our panel. Thank you, Joe. Amy, Sandra, and Joe, I'm very honored to have you on today's program. A warm welcome, and thank you for joining us.
Thanks, Matt. Great to be here. Looking forward to the conversation.
Yeah, thank you, Matt. It's great to meet Sandra and Joe. I look forward to today.
Yeah, thanks, Matt. I'm excited to take a break and looking forward to the conversation.
Excited to take a break from his vacation. Not the words of his, those aren't his wife words, those are Joe's words. Please thank her as well. Before we jump into the discussion, I want to implore all of you who have tuned in to squeeze as much value as you can out of the next 60 minutes. The best way to do that is to ask questions early and often. I promise you we'll get to as many of them as we can. So let's get started. Amy and Sandra, I want to start with the two of you. And I don't want to go too long on this, but I want to get a quick synopsis from each of you, just sort of for a level set and some context in the discussion around your respective ADC programs, and more specifically, sort of the key IP that those programs are built on in your organizations. And we'll get into the why that's of interest to me in a minute. But Sandra, maybe we can start with you. Give us a brief synopsis of what's going on there at Mythic.
Sure. I mean, as you said, ADCs have emerged as a really highly promising modality. Regulatory activity has been swelling, lots of approvals, and HER2 being the real standout. So that brings us where we are today, where when ADCs work, they work really well, but they are restricted, particularly in solid tumors, to really a small handful of targets, and within those targets, really to a subset of patients with the highest levels of expression. Really due to a narrow therapeutic index. Less than about 5% of any given dose actually makes it to the tumor cell, and the rest goes elsewhere, really increasing these unwanted toxicities. So we think this is really the problem, the limitation that ADCs face currently, and that's exactly what we're trying to do at Mythic, is to solve this with our fate control technology. So you can think of fate control as really a suite of tools and analytics that we've developed with a focus on the antibody component of the ADC. We're using this protein engineered focus to specifically enable us to increase the fraction of any given dose that's taken up in the tumor cell, where it's going to drive efficacy, and decreasing the uptake in normal cells, where it drives this unwanted toxicity. So these two aspects of our technology, we believe, give us the potential to go after tumors with lower expression levels of the target, which in turn will allow us to expand the population of patients who might benefit. So our lead program, as you mentioned, MYTX11, is in a phase 1 clinical trial. We're going after non-small cell lung cancer initially, really with the potential to treat other tumor types such as gastric and head and neck. This ADC is actually engineered to boost ADC uptake in the tumor cells by 3 to 5-fold, and at the same time decrease the uptake in the normal cells. Behind this, we have a number of programs in the pipeline and looking to nominate our second program and development candidate later this year. In terms of IP, we have multiple layers of patent coverage ranging from composition of matter, the antibody and the ADC, really to ring fencing patent applications such as new methods, formulations, and combinations. So all of this, our programs are built on Mythic developed and or Mythic owned IP rights.
Fantastic. Thank you, Sandra. And you addressed a number of the challenges that we're going to talk about a little bit later in the program. And that IP, like I said, where your IP lives is sort of specific to, we're important to this conversation. I'm going to get Joe's perspective on that here in just a minute about how, each sponsor that we see in this space seemingly tackles what their core is from a specific perspective, and then outsources like all sorts of stuff. Amy, what's the story at Vincerx in terms of the IP that you guys own, and sort of the I guess synopsis of what your ADC aim is?
Excellent. Yeah, thank you. So Vincerx Pharma is an early stage clinical biotech company. And we, like Mythic, we've really thought about what are the challenges with ADCs, and really started to think about how we can improve the safety and tolerability of the current ADCs. So we've built a platform we call Vincerx, and it really has kind of 3 major design solutions that we're putting into place that are important to solving these problems, but also that's where our intellectual property lies. So we've really been focusing on the lack and trying to avoid the bystander effect. And so we've been really carefully designing linkers. So the linker in between the antibody and the payload, and really making sure that is a very tight link, and cleavable where we want it to be cleaved. And so we've built a linker that is cleaved by a lysosomal protease called legumine, and it's only inside the cell. So we're only getting intracellular cleavage. And we think that's an important step in the safety and tolerability. The second is we've been building payloads. Really potent payloads with novel mechanism of action. A lot of the ADCs are kind of based on kind of 2 main classes of payloads, and so we're trying to expand that. And I can talk a little bit about kind of the novel mechanism of actions that we're thinking about, both from a kind of a targeted cytotoxic agent, and also even moving toward tyrosine kinase inhibitors, or very specific inhibitors that you might want to include depending on the indication that you're working on. And then the third kind of design solution is what we call our cell trapper. And this is actually a chemical moiety that we're building onto the payload to make the payload hydrophilic. And it gets hydrophilic, it's permeable, it gets into the cell, but it can't leave the cell. So you get accumulation of your payload inside the target cell. So I really think that this novel platform allowed us to identify our lead candidate, which you mentioned. It's a CD123 antibody-drug conjugate that has a novel payload, a kinesin spindle protein inhibitor. So that's currently in the clinic. It's in dose escalation for patients with AML, MDS, and BALL. In addition, we have a first-in-class small molecule drug conjugate, which is a little bit different. It's not using an antibody, it's using a small molecule. And we've designed that for multiple solid tumors. And we also have a best-in-class CDK9 inhibitor that's in the clinic too, but I won't talk about that because that's not an ADC. But you can kind of see we have both a small molecule and large molecule expertise within our company.
Yeah, very good. As you were speaking, Saurav just chimed in and asked you to just clarify. You mean the linker is cleavable only inside the cell? You mean tumor cells and not normal cells, correct? I'm assuming that is what.
So yeah, so legumine is concentrated in tumor cells, so that is giving us a little bit of selectivity. But like other ADCs, we're relying on that antibody, that targeted antibody, to bring it to the cancer cell.